DNA contains the blueprint for all life and is by far the densest information storage mechanism known in the universe. For example, the amount of information contained in a pinhead volume of DNA would fill a stack of books 500 times higher than from here to the moon. The program code and design of such an incredible system indicates a supremely intelligent designer.
And information itself is not derived from matter. Information always can be traced back to an intelligent mind. And if you believe that a code system could come into being without an intelligent mind, the onus is on you to prove that that's not true, because with all our observation, which is what science is mostly based upon, we're winning this argument. The genetic instructions don't write themselves any more than software programs write themselves. And decoding machinery requires energy from ATP, built by ATP synthase motors, built from instructions in the DNA, decoded by ribosomes. The simplest creature today is what they call a mycoplasma genitalium. It's got 580,000 letters coding for 482 proteins. That's 580,000 letters of information that you have to have anyway, and that's for the simplest thing we've ever seen.
Yes, there are changes, and some of the changes are even very large, but those changes don't lead to evolution, and even leading evolutionists agree with that.
Natural selection does not explain the origin of new variants, only the process of changes in their frequency.
How did new biochemical pathways, which involve multiple enzymes working together in sequence, originate? Evolutionary biochemist Franklin Harold wrote, "We must concede that there are presently no detailed Darwinian accounts of the evolution of any biochemical or cellular system, only a variety of wishful speculations."
One of the arguments for design is that some of the things that we see inside living things are irreducibly complex. You would have to have all the structures in place at once in order for them to function properly. Living things look like they were designed. So how do evolutionists know that they weren't designed?
Design is based on what we do know. We know, when we look at things, we can tell that things have been designed. You know design when you see it. Even though you don't know the design, you didn't see it be designed. Speaking of rocks in a circle, think of Stonehenge.
Right.
Nobody thinks that wasn't designed.
Just weather erosion,
yes,
random processes.
The wind moved those rocks into position.
The more I conducted my own studies, including a period of time at NASA Ames Research Center, the more it became apparent that there were multiple difficulties with the chemical evolution account. And further experimental work showed that amino acids do not have the ability to order themselves into any biologically meaningful sequences. We have not the slightest chance of a chemical evolutionary origin for even the simplest of cells. So the concept of the intelligent design of life is immensely attractive to me and made a great deal of sense, as it very closely matched the multiple discoveries in molecular biology. When you move into the area of intelligent design, although you argue to intelligent design through a dense field, if you will, of empirical data, and it's only after extended encounter with those data that you come out at the end with an intelligent design conclusion. Nevertheless, it makes many scientists uneasy because this is thought of, this causation, intelligent causation, as ultimately untestable by the traditional methods of science. So if you define science as excluding the possibility of intelligent cause at the outset, then any proposal that is made, however many data back it, is ruled out of court from the beginning. So,
in the 19th century, evolutionary scientists who followed in the wake of Darwin were convinced that life was exceedingly simple in its cellular form. The simplest form of life was exceedingly simple in its structure. The cell is a simple, homogeneous globule of plasm. Proteins do lots of important jobs in the cell. They catalyze reactions, they form the structural parts of molecular machines, they help to process and copy genetic information. They're really the workhorses of the cell. Now, fascinating what we know about the structure of proteins. The first three-dimensional structure was elucidated in 1958 by Kendrew in Cambridge. Sequence specificity. Something is sequence specific if the function of the whole is determined by the arrangement of the parts. Words are sequence specific. Sections of computer code are sequence specific. And proteins are sequence specific. This is an animation of how the digital information in the DNA directs the construction of proteins.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is often transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
So this is what's become of Huxley's undifferentiated protoplasm. The simple cell is no longer simple. Digital information is being used to direct the construction of mechanical parts. This is what's going on inside living systems. This has been part of the mind-blowing development that's called the molecular biological revolution. In biological systems, like in language and computer code, you have complexity, a lot of non-repeating, highly improbable arrangements of characters, but those arrangements are also specified to perform a function. Bill Gates puts it this way, he says, the DNA is like a computer code, only much more complex than any we've been able to devise. We have a striking appearance of design. Most proteins are many hundreds of amino acids long. The average protein is something around 300 amino acids. The protein, I have a colleague, Doug Axe, who's here with us tonight, who's done experimental work on very modest length proteins of a shorter length, about 150 amino acids. And if you begin to think about this mathematically, a very obvious problem arises for the chance hypothesis. If you have 150 sites with 20 possibilities at each site, you have 20 to the 150th power possibilities, which in base 10 computes to 10 to the 195th possibilities. So the odds of getting the probability of getting any particular sequence stands at 1 chance in 10 to the 195th.
We have never been able in any way theoretically to examine the central Darwinian claim that natural selection and random variation can account for a great deal of complexity. There is nothing like that in biology, in Darwinian theory. A kind of a canonical demonstration that this mechanism, random variation, natural selection is adequate to the generation of this level of complexity. I can program a computer with the equations of general relativity or with the equations of quantum mechanics, and I can say, all right, what are the consequences? I can actually see the consequences emerge in a simulation. We can't do any of this in biology. And that should prompt any reasonable person to ask, why not? If this is such a simple mechanism, it could easily be programmed on a computer, how come we can't set up a computer and create something of biological-like complexity? How come we cannot see the unfolding of an evolutionary process the way we can see the unfolding of an evolutionary process in physics? This is a very serious question. I've looked at all the genetic algorithms. I'm trying to write a genetic algorithm myself. And the sheer fact is, without a tremendous amount of very special manipulation and ad hoc constraints, the computer is not going to generate anything realistic if it uses Darwinian mechanisms, and it will generate something realistic only if it doesn't use Darwinian mechanisms. This is an important point. 50 years after the computer revolution began, we have a splendid tool for assessing the intelligibility and viability of Darwinian theory. And everything that we know, I think this is the uniform experience of anyone working in genetic algorithms, indicates these mechanisms will not work. They will not work for their intended purposes. And finally, there's the utter absence of laboratory evidence. I mean, random variation, natural selection, we should be able to start manipulating organisms. When we look at dogs, no matter how far back we go, it's dogs. When we look at bacteria, no matter what we do, they stay bugs. They don't change in their fundamental nature. There seems to be some sort of an inherent species limitation, and we have no good explanation for this in terms of Darwinian theory. We should have far more flexibility, far more plasticity under laboratory conditions than we actually do if Darwinian theory or anything like that were correct. What we see in nature, what we see in the laboratory, is very highly bounded variation, cyclic variation, as for example finch beaks in the Galapagos Islands. That's about all we see. Small variations. Why is that if Darwinian theory is correct? These are evidentiary points that I think need to be stressed, need to be examined openly, honestly. And they never are, of course. Never are.
His theories of prebiotic natural selection are problematic because, quote, they need information, which implies they have to presuppose what is to be explained in the first place. The sticks represent chemical bonds. Now, notice that there are chemical bonds between the sugars and the phosphates, chemical bonds between the sugars and the bases, but there are no chemical bonds between the individual nucleotide bases. Now, this has a really powerful implication for their self-organizational model, because if there's no chemical interaction here, you can't invoke chemistry to explain the sequencing. You can't even invoke the attachment site here, because this bond is called an N-glycosidic bond for people who are keeping track on their scorecards. This bond is the same in every case, so that at each site along the DNA chain, any one of the four bases can attach. It's chemically indeterminate. Any one of those bases can accept, or any one of those sites can accept any one of the bases. The chemistry, in other words, is not determining what attaches or the arrangement of the bases that attach to the medium. The message is here, the chemical message is here, the medium is here, but the chemistry is not determining the sequencing.
DNA is not simply a computer programming language, but as you have already seen, there are some startling analogies between DNA and computer code. We can view each cell as a CPU, or a central processing unit, running its own kernel. As you probably know, in computing, the kernel is the main component of most computer operating systems. It's a bridge between applications and the actual data processing done at the hardware level. So each cell has a copy of the entire kernel. But amazingly, each cell chooses to activate only the relevant parts, which modules or which driver it loads, so to speak. If a cell needs to do something or call a function, it calls up the right piece of the genome and transcribes it into RNA. The RNA is then translated into a sequence of amino acids, which together make up a protein for which the DNA coded. Now for the truly amazing part of the equation. This called up and coded protein is then tagged with a shipping address. This address is a marker consisting of several amino acids which tells the rest of the cell where this particular protein needs to be deployed. Once the protein arrives at its intended location, the delivery instruction is then stripped off and several post-processing steps are performed, activating the protein to now do its job. The protein is not activated until it arrives at its marked and intended location. The amazing and complex intelligent communication functions of DNA are myriad.
Can you give an example of a genetic mutation or an evolutionary process which can be seen to increase the information in the genome?
Can you just stop when
Yo, block off. I ain't karate kidding. Bruce Lee.
I told you we don't know.
So you have no idea how it started?
No, nor has anybody.
Nor has anyone else.
You can't hurt me. You can't hurt me. You can't stop me. You can't drop me. You can't block me. You can't break me. You can't shake me. Gonna make me. This is the takedown. This is the takedown. This is the takedown. This is the takedown.
The God of the Old Testament is arguably the most unpleasant character in all fiction. Jealous and proud of it, a petty, unjust, unforgiving control freak, a vindictive, bloodthirsty ethnic cleanser, a misogynistic, homophobic, racist, infanticidal, genocidal, filicidal, pestilential, megalomaniacal, sadomasochistic, capriciously malevolent bully.
Well, then who did create the heavens and the earth?
Why do you use the word who? You see, you immediately beg the question by using the word who.
Well, then how did it get created?
Well, by a very slow process.
Well, how did it start?
Nobody knows how it started. We know the kind of event that it must have been. We know the sort of event that must have happened for the origin of life.
And what was that?
It was the origin of the first self-replicating molecule.
Right, and how did that happen?
I told you we don't know.
So you have no idea how it started?
No, nor has anybody.
Nor has anyone else.
You can't stop me. You can't drop me. You can't block me. You can't break me. You can't shake me. Gonna make me. This is the takedown. This is the takedown. This is the takedown. This is the takedown. Sweep the leg. Sweep the leg. Sweep the leg. High tech defense mechanism.
And I look at the attempts to explain the origin of information based on chance, based on necessity, based on the combination of the two, and I infer that intelligent design provides the best explanation because each of these other forms of explanation have been shown for various reasons, either empirical, theoretical, or both, to be incapable to produce the information necessary to produce the first life.
Yo, block off. I ain't karate kidding. Bruce Lee, giving you double vision, no fear, no mercy. Gloves off, try and hurt me. You can't hurt me. You can't hurt me. You can't hurt me. You can't stop me. You can't drop me. You can't block me. You can't break me. You can't shake me. Gonna make me. This is the takedown. This is the takedown. This is the